Hello and welcome to this episode of the VJNeurology Podcast. We recently attended the American Academy of Neurology annual meeting in Chicago, where many key trial updates across various neurological conditions were presented. This podcast will feature some of our top picks from the conference. We begin with some trials exploring orexin receptor agonists in narcolepsy. Emmanuel Mignot will kick us off with the updates on oveporexton and ulixacaltamide, followed by Bruce Corser, who shares findings from a phase 1 trial of the orexin agonist E2086. This podcast is part of the Sleep Disorders channel on VJNeurology, which is supported through educational grants from Takeda and Alchemist. These supporters have no influence over the production of the content. This podcast is intended for healthcare professionals only.
So I think it's a very exciting time for narcolepsy because of this orexin agonist. As you know, it was discovered that the cause of narcolepsy is a lack of orexin, which is due to an autoimmune process, which is very likely triggered by specific flu strains. And it's a lifelong disorder, and people lack the orexin. And for a very long time, the treatment of this disorder was really symptomatic. We would use drugs that will knock out the patients at night, like oxybate, sodium oxybate, which makes them sleep very deeply, and then that helps them to be more awake during the day, or stimulants or antidepressant to suppress REM sleep symptoms. However, recently, to my delight, there have been a development of orexin agonist, which can replace what is really missing, which is the orexin peptide, which is the cells are missing as a result of the autoimmune process. And of course, as you can expect, these drugs are spectacular in this form of narcolepsy where the orexin is missing. It's a little bit like insulin for type 1 diabetes. And we're seeing now the results of many of the trial. One of the drug, oveporexton, is going to be probably coming out in six months to a year or 9 months. So it's relatively soon. If the FDA approve it, and I'm not, of course, I can divine what's going to happen. And then the other one that's following up from Alchemist is probably about a year or so, but maybe a year and a half behind. And both of these drugs, so one of them just completed phase 3, and the Takeda drug, and the results are really amazing. What you see is a reversal of the symptoms of narcolepsy in a way that we have not seen before.
So Eisai's study evaluated E2086, which is a new orexin 2 receptor agonist. This is for treatment of narcolepsy. Specifically, this study involved type 1 narcolepsy, that is patients with narcolepsy and cataplexy with orexin deficiency. So the study was a 5-way crossover study involving a single dose of either modafinil, placebo, or 3 doses of E2086. So this study looked at various doses and compared these doses with placebo or modafinil, which is a standard treatment for narcolepsy. And what was found was that all of these doses separated from placebo in terms of promoting wakefulness, and this was identified with a maintenance of wakefulness test, which is a standardized method of objectively determining the degree of excessive daytime sleepiness. And furthermore, all the 2 higher doses separated from modafinil in terms of subjective sleepiness. This is evaluated by the Karolinski sleepiness scale, which is also a well-recognized method of evaluating subjective daytime sleepiness. So this is an important study in that it demonstrated that these several doses of this new orexin agonist substantially separated both from placebo and from existing therapies for treatment of narcolepsy type 1. And this is an emerging therapy that's going to be very effective, and not only for people with type 1 narcolepsy, but also eventually for type 2 narcolepsy as well.
We now move on to some updates in movement disorders. First, you will hear from Victor Sung, who discusses the 3-year outcomes of a phase 1-2 study exploring a single dose of gene therapy AMT-130 for Huntington's disease. Jill Farmer will then share top-line results from the Essential3 Study 2 of ulixacaltamide for essential tremor.
So just as a way of introduction, the AMT-130 study, remember, is a single-dose gene therapy trial. All of the patients that are presented, they had their surgery to implant the drug into their striatum over 3 years ago, and the presentation is on the patients that have reached the 3-year follow-up data. And the first thing is it's a safety study. So the most important thing from a safety standpoint is there are no new safety events. There are no new neurologic problems or things coming up at the 3-year mark. So that's important to note, as gene therapy is invasive, and, continued safety or no new safety things is a good sign. And then in particular, the results that have made a lot of buzz are the clinical results for these patients. So all of the patients were unblinded at a year. So in the 3-year analysis, we're comparing to an external matched control group. And compared to that external control group, the treated patients are relatively stable, whereas the external control group are following the natural progression of the disease. And so at the 3-year mark, this continued further separation from the control group. And so the treated patients are significantly less progressed than the external control group. And when you compare the differences in how much they progress, it's about 75% less progressed. So that's great. And it's on a composite unified Huntington's disease rating scale measure. A composite measure combines motor, cognitive, and functional measures in there. And then the other detailed part of it is that in a composite measure, you can sometimes hide something negative with something positive. So it's important to note that the individual subscales of the composite measure are also all positive, favoring the treated patients. So that's, again, a positive result. And then the last major result that's being presented is the CSF neurofilament light chain data, which neurofilament light is a known biomarker for progression in a lot of neurodegenerative diseases such as MS, ALS, and is increasingly accepted in Huntington's disease as well. Normally increases over the lifetime of a patient with neurodegenerative disorders. And in this trial, the patients had about this flat or slightly decreased NFL levels at the 3-year mark compared to what we would think should be an increase over time. So those are the summary of the findings of the AMT-130 trial that are being presented here today.
The Essential3 Study 2 was one of 2 studies conducted in parallel through Praxis Pharmaceuticals. And what was interesting about these studies is that they were a decentralized study, meaning that there was no brick and mortar site for the patients. The patients were seen in their home with a study nurse that went out to the house and then evaluated by investigators through a telemedicine visit at the same time as the nurse being there. It was unique in that we had participants from all over the country, all 50 states, and we had about 200,000 patients self-register out of interest for the study, which goes to show the need for a treatment for essential tremor in the United States. And of those 200,000 patients, through inclusion criteria and rigorous evaluation, they were whittled down to about 698 patients. Those 698 patients were then consented to both studies. They didn't know if they were going to be enrolled in study 1 or study 2. They were randomized in a 2-to-1 fashion. And then in study 1, it was your typical study. They had a placebo and drug, and they were just followed over the course of 14 weeks. Study 2 was the same study essentially from the first 8 weeks, except everyone received the drug. And then at the 8-week mark, they were randomized in either continuing on study drug or withdrawing from study drug. And the main focus was looking to see if their symptoms returned as the study drug was removed. And the symptoms did, so it met its endpoint. I think it has a huge potential for impact. And the reason is, in the United States, we only have one FDA-approved medication for essential tremor, and it's not even a neurological medication. It's borrowed from cardiology. It's a beta blocker called propranolol. And we do treat with other things, but all of those other things are borrowed from other aspects of neurology. There's anti-seizure medicines and anti-headache medicines and neuropathic pain medicines that we try to use for essential tremor treatment. And unfortunately, patients don't stick with our current treatments available because efficacy is lacking and tolerability is also a problem. Side effects of, making people feel dizzy, lightheaded, cognitively, medicine heady sort of a thing. So this is the first drug that is really looking at the pathophysiology behind essential tremor, and it's focusing centrally in the brain on the circuit that drives tremor. And it's blocking the receptors in the brain that drive the tremor, hoping to sort of reset the circuit so that there's less tremor and smoother movement wherever the tremor happens to be on the body, head, arms, legs, neck, that sort of thing. So since there's nothing readily available, to have something that is pharmacologically, mechanistically appropriate that has been shown to be effective is very exciting. It's also been shown to be well tolerated in the studies, so it has the potential for very strong clinical utility.
Kinga Tomczak speaks next on a phase 3 trial evaluating the efficacy and safety of ecopipam for Tourette syndrome.
So I will start with introduction of this compound called ecopipam. It is the first in class selective dopamine 1 receptor inhibitor. So far, as I mentioned, we were using D2 inhibitors, so this antagonists. So this one are modulators, and this one is targeting a different receptor in basal ganglia. And what we've learned in the previous phases, is that it seems to work better than placebo. In phase 2B trial, in a pediatric population, it showed that there's at least a 30% improvement in patients on the drug compared to placebo, which was statistically significant. And patients who were in the open label extension afterwards, they even had an improvement of 40% at 12 months. So this trial that I was presenting at AAN is actually looking at efficacy and safety tolerability of this drug when it's given for longer time. And the way the trial was designed was that patients initially, everybody who was enrolled and was at the baseline visit was getting the medication. So the first part was called open label. All the patients were exposed to the medications, and many of them had an improvement, but they had to maintain the improvement at week 8 and at week 12. So for a whole month, they had to have a clinically meaningful improvements, which is more than 25%, more or equal of 25% of the tic severity that was seen initially at baseline. And at this point, there was a randomization. So we randomized patients into a ecopipam arm and placebo withdrawal. So they were getting less and less medication for a couple of days or weeks, depending on their weight. And then they remained randomized for as long as they had so-called relapse. And relapse was defined as they were losing half of their benefit that they were getting at the randomization. And so then we were looking at how long did it take for patients on ecopipam compared to placebo to relapse. And what we found, which was really exciting, is that patients for 12 weeks of duration of this part of the trial, patients on ecopipam were not really reaching this 50% threshold. So they continue to have some benefit and compared to the placebo. And so we looked at what is called a risk reduction, and they had 50% risk. Patient on ecopipam had a 50% risk reduction compared to placebo, and this was clinically statistically significant. And then the secondary outcome measure, the secondary endpoint was to look at the whole populations of pediatric and adults, and we saw similar trend, similar results. So that was also very reassuring. In terms of the side effect profile, because that's really important, especially that this is one of the few trials, specifically in mostly pediatric patients. We always worry about serious adverse events. And thankfully, there was hardly any serious adverse events. There were some mild side effects, such as patients experienced somnolence, especially initially some fatigue, or some patients also had insomnia, which with time in many have improved. And there was some anxiety or depression that we saw at this trial. But what is really important is that we did not see weight gain or metabolic side effects, and we did not see these movement-related adverse events that we can see in some other drugs. So there's definitely an unmet need to find medications that will be effective and not have serious side effects. And when we looked at this trial, as I said, the results are very promising, but it has to go through the FDA approval. And currently it is being actually under an FDA commission process. It will take several months, and it's hard to know when the drug will be available. We hope maybe at the end of 2026, but it could be 2027.
There were many key clinical trials in headache presented at AAN. Peter Goadsby outlines some of these trials next.
During the recent American Academy of Neurology meeting in Chicago, there was a trial session, presented some of the most interesting things that are happening in the clinical trial arena. And I'm just going to pick a couple of them out. First one I'll pick out, and I have conflicts of interest with these because I'm interested in these, and I've certainly advised the companies. I want to say that straight up. The first one I'll highlight, because it's entirely new, is a drug called Elismetrip, Elismetrep. If you've never heard of it, you probably haven't. It's a TRPM8 receptor antagonist. Now, the TRPM8 receptor is the cold receptor. If you take menthol and put it on your head and it feels cold, it's because you're activating that receptor. So what was done was a double-blind, placebo-controlled, randomized dose-ranging study, 2, 5, 10, and 20 milligrams of Elismetrip in the acute treatment of migraine. About 30%, a third of the folks in the study were triptan resistant, meaning having failed at least one triptan, and about a third of them were on a preventive. The primary endpoint was 2-hour pain-free for the 20 milligram dose. And it's worthwhile saying that's a modest dose in the context of this drug, which had been developed in another indication that didn't go forward, not in migraine. And there's over 1,000 people who've already been dosed with this drug, so it's really quite de-risk in terms of safety. So the primary endpoint, 2-hour pain-free, 9.1% for placebo, 17.6% for the active. This wasn't significant, and the primary endpoint was done using an electronic diary. By making inquiry of one patient who didn't fill an electronic diary in 2 hours but filled it in earlier and later, the 2-hour pain-free rate was 19.6%. Problem of relatively small numbers, although there were nearly 400 patients in the study across all of the 4 doses and the placebo, and that was significant against placebo, which was at 9%. Without any sort of adjustments, the 2-hour most bothersome symptom rate, 24% versus 38% for active, was significant. These are nominal p-values. And the 2-hour pain relief data, 42% versus 59%, was also significant. And pain relief is quite important because it correlates quite tightly with improvement in disability, improvement in function. So, entirely new entity, the TRPM8 receptor. Certainly have to watch what happens with that. Quite importantly, from a practice point of view, it's been very difficult to get studies sorted out with the pediatric and adolescents. And Dr. Bryson presented a study, the 2 studies in children and adolescents in episodic and chronic migraine with fremonizumab. These are phase 3 studies. Patients were 6 to 17 years old. The standard differential diagnosis in the sense that episodic migraine 14 days or less and chronic migraine 15 or more days. They were randomized 1-to-1 with fremonizumab and placebo. Importantly, there was dose adjustment, so 120 milligrams if the participant was under 45 kilos and 225 if they were 45 kilos or more, took placebo or active for 3 months. And the primary endpoint showed that for episodic migraine, there are 234 patients in the episodic study, 289 participants, I should say, in the chronic study. For the episodic study, fremonizumab beat placebo. And for the chronic study, it didn't, which is great for those with episodic migraine. It's great to see a pediatric adolescent study positive. It's I have to say, I thought the audience thought, and I'd have to join them, it's a bit mystifying why it's difficult that the chronic migraine study was not successful. All of that said, we had a positive randomized placebo-controlled trial in episodic migraine in children and adolescents. An interesting study that was presented tried to look at the cognitive toll, you might say, and using real-world evidence in what was called a MIND cohort, MIND, so migraine impact on neurocognitive dynamics. Dr. Zadi presented this, and they enrolled 129 adults with migraine via social media, and for 30 consecutive days got them to record their daily headache status, and they did 3 brief smartphone cognitive tests: a simple search test for attention and processing, color dots for item localization, visual working memory, and color shapes, feature binding detection in visual working memory. And they looked at the performance across time. This is a pretty typical cohort of 38-year-old, predominantly females. And what they showed was on headache days, on the symbol search, there was a reduction in response times compared to their baseline, and similarly on the coloured dots, but not on the coloured shapes. And the first 2 were significant. It's quite remarkable, and I think it's been underappreciated by physicians, but certainly it is appreciated by patients, that there is a cognitive penalty to migraine, and that those cognitive reductions were tracked with the attacks. And you can do that in an objective way. And it goes to show you more broadly that this use of real-world digital endpoints, real-world digital technologies will enable us to get some very important insights into what's going on a day-to-day basis in people with migraine.
Next, you will hear from Amy Waldman, who shares findings from a phase 3 trial evaluating zilganersen in Alexander disease.
Pathophysiology of Alexander disease has been very well described, including the genetic variant in GFAP, which is the name of the gene that produces glial fibrillary acidic protein, which is a protein that accumulates in astrocytes. With that accumulation of protein in astrocytes, there is a further buildup of fibers called Rosenthal fibers throughout the brain, but it's not exactly known how those fibers disrupt brain function and cause patient symptoms. There are actually no approved treatments for this disease, so we really are only left with the option of treating symptoms. For example, using anti-seizure medicines in the subset of patients that have seizures, or using medications to help with tone or spasticity, or those to help with some of the GI side effects in this disease. Zilganersen is an antisense oligonucleotide, or ASO, that binds to the GFAP RNA, and by doing so, causes the body to make less GFA protein. So ultimately, the goal is to reduce the amount of protein being made to slow or halt disease production. In this study, 49 patients were randomized in total, beginning at the start of the study with 8 participants in the initial phase 1 cohort of 25 milligrams compared to the control. And ultimately, 24 participants were randomized in the phase 3 or the 50 milligram cohort. And across both cohorts, there were 17 control participants. And over the course of the study, there was a stabilization of gait speed in those patients who received zilganersen at week 61, whereas there was a steady decline in motor skills in the patients receiving control. There were also a number of other outcome measures that favored zilganersen treatment that really covered the gastrointestinal, other motor symptoms, cognitive and autonomic symptoms in this disease. The safety profile was generally tolerable and acceptable. There were some mild to moderate adverse events, and serious adverse events occurring in the zilganersen group actually occurred at a frequency that was decreased compared to the pooled control, and generally consisted of seizures, vomiting, and scoliosis, some of which was deemed to be related to the underlying condition rather than the treatment itself. This has been a major milestone for this disease community and a major advancement in having a potential first disease-modifying therapy. And in the future, it'll be exciting for us to learn more about the different presentations, how zilganersen can halt or stop disease progression, and ultimately how we improve how patients are feeling who are suffering from this disorder. I would just like to thank the entire disease community, the patients that were participating in this trial, but also all of those participants who visited me at CHOP, helped me to understand this disease in incredible detail. And it would not have been possible without the tremendous efforts of the entire Alexander disease community.
Several key trials, including the Hercules trial, have shown the efficacy of tolobrutinib in multiple sclerosis. Patricia Coyle will now give an overview of this trial and discuss a subgroup analysis that was presented at AAN.
Hercules study was a phase 3 study that focused on non-relapsing secondary progressive MS. It was a randomized, placebo-controlled, double-blind, event-driven study. It entered 1,131 individuals. They had to have been free of any clinical relapse in the prior 2 years and had to have progressed in the year prior to entry. They ranged in age from 18 to 60 with an EDSS of 3 to 6.5. The primary outcome was 6-month confirmed disability progression that was significantly slowed in favor of the BTK inhibitor, a 31% decrease in progression with the oral BTK inhibitor. The interesting thing in the 2 phase 3 relapsing trials that read out at the same time, they failed because there was a direct comparator, teriflunomide, and the primary outcome was annualized relapse rate, and it didn't beat teriflunomide, although the annualized relapse rate was low across the board. But when you looked at confirmed disability worsening in this relapsing cohort, the graph separated in favor of the BTK inhibitor. So even though you can't count that because the primary outcome was negative, it really indicated that you were likely seeing an effect on PIRA in those relapsing patients, confirming an impact on progression in MS. There's always a concern in studies around the time of the pandemic and then with the Ukraine war, et cetera, where they recruit from outside the United States, and there are not a lot of North American individuals that participate in this study. In Hercules, this was the case. North Americans made up about 14.3% of the patients entered into that study of a BTK inhibitor versus placebo in non-relapsing secondary progressive MS. About 2/3 were from the United States and 1/3 from Canada. And indeed, you saw even in this subset analysis, and of course, the numbers are more limited. There were 162 cases that were entered into this subset analysis. But if you look at it, they certainly had a benefit in less likely to develop the 6-month confirmed disability progression. In fact, that was 54% lowered compared to 31% in the overall study. And if you looked at the United States, it was 62% decrease. So you really had a strong impact in favor of the BTK inhibitor. The other interesting thing was that in this North American cohort, only 4.4% had contrast lesions at entry compared to 12.7% in the whole study. Yet those patients had a very strong impact on progression. Now, normally, contrast-enhancing lesions would be a relapsing component, and you would think you might get a relapsing treatment response, not a true progression treatment response. But this study kind of gave data saying that you were indeed seeing a true progression impact.
Finally, we hear from Daphne Amado, who shares updates on gene therapy-based approaches for the treatment of ALS.
I'm a scientist, so I run a lab, and we're involved in developing gene therapy approaches for ALS. So I'm very immersed in this world. The gene therapy as an approach for ALS, the power of that strategy is the ability to reach the relevant cells in the nervous system that need to receive a therapeutic. So I personally work with adeno-associated viruses, or AAVs, to deliver therapeutics that can reach the affected motor neurons in the brain, brainstem, and spinal cord. And other groups are achieving that through different strategies like antisense oligonucleotides, and people are working on lipid nanoparticles and other delivery methods. In terms of what is the therapeutic being delivered, there's a lot of different approaches. So a lot of gene therapy targets a genetic mutation. So if people have variant in a gene that's associated with ALS, you could deliver machinery to edit that gene or to deliver a working copy of that gene or to knock down that gene depending on what problem that gene is causing. Additionally, there are some shared downstream things like TDP pathology that can be modulated. So what my lab works on is targeting that downstream pathology, in our case via reduction of ataxin-2, but there's other approaches that we're investigating and undertaking to unclump TDP-43 and restore it to its nuclear location where it can do its proper functions. So there's all kinds of different ways that gene therapies are being employed in ALS. So the strategy that's probably the furthest along clinically is antisense oligonucleotide therapy. So these are stretches of nucleotides that are designed to pair up with DNA or RNA in a cell and modulate its function. So ASOs can knock something down. It can target an RNA for degradation, or it can mediate alternative splicing and correct the function of something. And so one of the therapies that is actually FDA approved is an antisense oligonucleotide that targets SOD1. SOD1 variants are associated with about 2% of all ALS. And for those patients with that disorder, receiving monthly intrathecal injections of tofersen, an SOD1 targeting ASO, extends lifespan significantly. It slows down the disease progression. And now the long-term data is suggesting that about a quarter of patients on that drug stop progressing, and a handful of patients have even shown improvement. So that really shows the power of a gene therapy approach if you are able to target the pathology effectively. There are a lot of therapies in clinical trial right now that are gene therapy based and that are targeting individual genetic causes or targeting the more general broad pathology in ALS. As I mentioned, when TDP-43 is not doing its job in the nucleus, a lot of things are spliced incorrectly, and you can actually lose certain proteins that your cell is meant to have. And so some of those proteins are stabmin2, UNC13A, and others. And there are gene therapy trials aimed at restoring those, delivering working copies of those genes to offset the pathology seen in ALS.
Thank you for listening to this episode of the VJNeurology Podcast. Head over to VJNeurology.com to see our full coverage of AAN. Be sure to follow us on X at VJNeurology and subscribe to our podcast on Spotify, Apple, or Podbean. Until next time.